respiratory rate, and the partial pressure of CO2 goes up. As the partial pressure of CO2 goes up, the concentration of hydrogen ions goes up. So this is a very quick kind of mechanism. This ventilation is pretty rapid responses. We can alter CO2 levels pretty quickly, and therefore alter hydrogen levels pretty quickly, and keep us in the. So over the course of the day, after you've eaten, things like that, your respiratory rate actually changes pretty rapidly to allow for this kind of compensation. If you were to just drink a lot of orange juice, very acidic, right? Can you tell me, do you think your respiratory rate might change before you drank the orange juice to after? Yeah. What should it do? Go up or go down after you drink the orange juice? Goes up, right? You've got too much hydrogen, you need to get rid of some. So we lower our respiratory rate, or increase our respiratory rate to blow off some CO2. So we have that kind of quick mechanisms, quick abilities to do those things as a result. Now, over long term, and actually at about the same time, but it just takes longer for this to occur, we can also do renal compensation. So this is all respiratory compensation. We also do renal compensation. And by renal compensation, we know that the nephron can selectively reabsorb or secrete ions, right? And the nephron can selectively reabsorb or secrete hydrogen, but also bicarbonate. Bicarbonate's the buffer, right? And so we could either conserve bicarbonate to help us buffer against acidic conditions, or excrete bicarbonate because we don't need the buffer if we're alkaline. So this renal compensation is by this mechanism. Now there are direct hydrogen ion transporters, and you can directly excrete or reabsorb hydrogen. And that happens. But the most common way is indirectly by changing the rate with which this buffer is retained by the blood in the urinary system or released in urine. And it also takes advantage of this carbonic anhydrase system. So this carbonic anhydrase system is not only used in red blood cells to help us transport CO2. Carbonic anhydrase is also found in cells of the renal tubule. Okay? So in the collecting duct, so remember the structure here, proximal convoluted tubule, descending limb, thick ascending limb, distal convoluted tubule here, and then it empties into collecting duct. And as this collecting duct passes back down through the renal medulla, we mentioned that ADH regulates the permeability and that helps us make dilute versus concentrated urine. But we can also regulate the pH of urine in that collecting tubule as it passes back down through there. And there are cells that line, specialized cells that line the collecting duct that are called intercalated cells. Now the word intercalate you've seen before with intercalated discs, right? Intercalate just means goes between. So these are cells that just go between the sort of cells that line the tubule. And these cells produce carbonic anhydrase. So these cells, these intercalated cells, have carbonic anhydrase. Okay? Now because they have carbonic anhydrase, this reaction will always be occurring. Right? Is there always water inside of a cell? Absolutely. Is there always CO2 inside of a cell? Absolutely. Now if you don't have carbonic anhydrase, it just stays like that. The CO2 comes out of the cell, gets picked up by the blood, transported into by diffusion into red blood cells, and then this is where that reaction would occur. But if this cell has carbonic anhydrase, as soon as those two are in there, this reaction occurs right here. It makes carbonic acid. Carbonic acid dissociates into the hydrogen ion and the conjugate base here, which is the bicarbonate buffer. You all right with that? So we have water, carbon dioxide, carbonic anhydrase. We're going to have hydrogen and bicarbonate. So this always is present here. Now this is very quick because this is always present. If the pH is in balance as things come down through here, no big deal, right? We'll just keep them present there. We don't need to excrete one over the other. But if we have the blood here under a state of acidosis, we want to keep one of these and eliminate one of these, right, to help us balance it. So if the blood has an excess of hydrogen, this could happen. And this does happen, but this takes time, right? I pump the hydrogen out of the blood into the interstitial space. I pump the hydrogen from the interstitial space into the intercalated cell, or not the intercalated cell, but just a cell. And then that comes through here, and then I pump it from this membrane here out into the urine. That takes a long time. How many times do I have to move it? 1, 2, 3 times to get it out here. So, and that happens. That's what we call direct maintenance of hydrogen ions, right? You directly move a hydrogen ion. Indirectly, I can affect it by this much more quickly. If this cell senses that the pH is acidic, and we have in this cell a buffer and a hydrogen ion, this cell just transports the buffer into the blood there, right, and takes the hydrogen ion and transports it into the urine right there. That's very quick. Right? It can conserve the buffer. So how does this help pH? Because this grabs a hold of the hydrogen ion, lowering hydrogen ion concentrations and bringing the pH back up to normal ranges. What happens to the pH of the urine? It's going to go down as the hydrogen ions go up. Okay. So that's very quick. It happens very quick. Similarly, so that's a type A cell. Type A cells, you can imply A functions in acidosis, would allow that to happen. So a type A cell under acidic conditions pumps a bicarb into the blood and excretes a hydrogen ion in the urine. There are many more of these cells here than there are type B cells. Now what would you expect a type B cell? B cell is going to function under alkaline conditions, so basic conditions. It's going to do the opposite, right? So a type B cell is going to pump the buffer out and the hydrogen ion in.